Hello, students. Welcome to the lecture 4 of recombinant DNA technology. So in our previous lectures, we have seen the process or the steps that how to do a molecular cloning, how to prepare the DNA and the vector, and then ligate them, and then what are the processes we can transfer to an host organism. In most cases, it is a bacteria. And then the processes of screening, if you can recall, it's mostly that firstly the antibiotic resistance, and then we can do the blue and white screening, which is also known as alpha complementation. And most popular and the simple one is the colony PCR, and the quick one obviously is the colony PCR, which you require either a insert specific or a vector specific or a orientation specific primers, which one is primer for the insert and one primer for the vector. And in that way, you can actually get some of the colonies of bacteria that have the expected clone of our DNA. But we always need to confirm these either by restriction digestions or by DNA sequencing. So the restriction digestions, as we discussed in previous classes, is just using the restriction enzymes. And when we know the sequence of the DNA, the whole plasmid or the fragment of DNA, we can check what are the enzymes is cutting or the cleaving that DNA in which position. So when we extract the plasmids, so the plasmid extraction, if you can recall in the first lecture, is how to extract the DNA. So this is the same procedure. There might be a little bit of difference between the plasmid extraction and the genomic DNA extraction. You need to check that one. But when you extract the DNA, you got the DNA and then you digest with your enzyme, which you have to find out before you proceed to the next step. That is digestions with the enzyme. So you digest with your expected enzyme, and you will know from the application you will use eventually that if the enzyme cut the plasmid in that specific position, what are the fragments you will get? And this can be identified by gel electrophoresis, which we have already discussed a bit, but we'll discuss in the next slides. And also the Southern blot analysis. So this is, as I've said previously, this Southern blot analysis is mostly used to check the copy number of expected or the transformed DNA into the host genome. How many copies are actually integrated into the chromosome of a host genome? It's more of a later stage experiment rather than checking just the plasmid. But DNA sequencing, which is very important to confirm that, okay, we get the plasmid, we extracted it, the restriction digestions looks very fine, we get our expected fragments, but still, if we proceed for the important experiments, like if you're preparing a DNA library or anything like that, you definitely go and check with the DNA sequencing, because it is possible that the DNA fragment you want to clone, it can be one or two base pair can be altered inside the bacteria. So it's need to be really checked and confirmed by DNA sequencing. Okay, we have got the right DNA fragments which we are looking for. So restriction digestions, as I said, that it's mostly determining the size and the orientation of the clone fragment. So in this way, as I just described now, that we actually know the plasmid as well as the DNA fragments. That is the map of the total plasmid. So this is the map of the PUC19 plasmid. There is no DNA fragment has been added. This is just the map of a this vector. And here we see that the SCA1 enzyme, restriction enzyme, has been cutting in 2177 base pair position in here. There is EcoR1 here, ND1 here, and BGL2, BGL1 also cutting in here. So and there is a multiple cloning site, and you see a lot of enzymes are here, which is cutting in here. So this is the position which we normally use to clone our DNA. So as the name suggests, this is called a multiple cloning site. So with the map, we know that what are the restriction enzyme is actually cutting the vector as well as the DNA, if we have the DNA in here, DNA fragment. So in that way, when we digest the plasmid, that is vector plus the DNA fragment, we will then know that, okay, these are the sites that has been cutting by these enzymes, and we will digest the plasmid with those enzymes, and then we are expecting these fragments to be present into our gel. So this is the process by which we can actually confirm a plasmid after molecular cloning. With the restriction enzymes, we've seen that there are like 3 types of restriction enzymes, but the type 2 is the mostly used into the laboratory and into the biotech companies. But there are definitely uses of the type 1 and type 3 as well. But there are some important thing about the restriction, that some of the restriction enzymes, they can just have a single restriction site into the plasmid. So they just cut and make a single fragment of the whole plasmid. But some of the restriction enzymes, they have a multiple cloning restriction sites into the plasmid. So they can actually have 2 or 3, or sometimes maybe 4. So they can cut the fragments into 2 or 3 fragments. So we need to get this information before proceeding to the restriction digestion step. And we need to know that which position, which enzyme is cutting, and what would be the band size. So the gel electrophoresis, after you doing the restriction digestions, you need to do a gel electrophoresis, which we have discussed previously, which mainly separates the DNA fragments by their size. And the samples are really running from negative fold to positive fold, that is cathode to anode. So in this experiment, this is just a vertical gel electrophoresis showing that, but mostly nowadays it's like a horizontal one. I'll try to put a video for that one. So we need one DNA ladder. So what is the DNA ladder? So this is actually a commercially available DNA fragments that give a specific size, that they're already producing this DNA with a specific size. So when you're running a gel, it will give a band in a specific size, like a 10 kilobase pairs, 8 kilobase pairs, 6, 5, 4, 3, 2.5, 1.2. So they have different kinds of DNA ladder, but mostly the 1 kb plus DNA ladder has been used into the lab. So you run the gel with the DNA ladder and with your samples. And if you can recall, for running your samples, you always run 2 samples, one which is untreated, that is, that sample has not been digested with the restriction enzyme, and the other one has been digested with the restriction enzyme, so that you can compare the same samples, treated and untreated, and you can compare the size, which is a reference, the DNA ladder. So DNA ladder gives you a reference that what size is this band. So in this way, we can actually identify uh, or confirming our molecular DNA fragments has been cloned to the vector.